Hey biologists, Mr. Fod here. Today we're going to be talking about cell respiration. First up are the need-to-knows. So first off, we're going to talk about the summary equation of our cellular respiration, including the source and ultimate fate of the reactants and products, differences between fermentation and cell respiration, the role of glycolysis in oxidizing glucose to 2 molecules of pyruvate while releasing some free energy in the form of our energy molecule, which is ATP, and how that pyruvate is moved from the cytosol into the mitochondria and introduced to our second phase, the citric acid cycle. First up, here is our general equation, and notice how we actually have both of these types of equations. We have both our cell respiration as well as photosynthesis. So comparing these two equations, they're actually very similar in that they're going to be using the same four ingredients. We have our glucose, C6H12O6, plus 6 molecules of oxygen, forming our 6 molecules of water, plus 6 molecules of carbon dioxide. In photosynthesis, once we get to that topic, it's going to be basically the same thing, but in reverse. And generally speaking, when talking about these chemical reactions, they're either going to be one of two things, either what's called exergonic, the energy is exiting, if you will, or being released. Notice how the ATP and even some heat energy is in the product side, thus being released, it's being produced here, versus photosynthesis is what's called an endergonic, the energy is entering or requires energy. Thus, our energy source, or sunlight in this case for photosynthesis, is going to be in the reactant side. So these are our two main equations that over the next several weeks that we are going to be focusing on. There are three main stages to cell respiration. First up is what's called glycolysis, and that is how you pronounce it, or if you want to be a little more fancy, you could say glycolysis is another way that you can kind of talk about it, and I'll explain why you could also refer to that as well. Second is kind of a two-parter. It's like 2.1 and 2.2 kind of thing. We have pyruvate oxidation as well as feeding into the citric acid cycle, also known as the Krebs cycle. Last but not least is going to be our oxidative phosphorylation, aka our electron transport chain, ETC for short, and chemiosmosis. So this, in a nutshell, is a fantastic diagram which basically talks about all the different stages. Our stage 1, glycolysis over here on the left, glucose 2 pyruvates, and then phase 2, again, this is kind of a two-parter here in the center, the pyruvate oxidation, including our acetyl-CoA, and then moving into the citric acid cycle. Last but not least, our phase 3, dumping into our oxidative phosphorylation, getting into the electron transport and chemiosmosis. First up, we're going to talk about glycolysis or glycolysis. So in its name, glyco referring to glycogens, which are stored sugar molecules, and lyse means to split apart. So the name literally means, in its translation, is the splitting apart of sugar molecules. And this is why when we start with our glucose, we are breaking it down or splitting it apart. Now, part of this, where this is actually going to take place, if you picked up on the last slide, is that this is not necessarily even require the mitochondria. So this is actually still in our cytosol or our cytoplasm, if you will. So it's not actually inside the mitochondria yet. So part of this is believed to be part of our ancient, these really early prokaryotes in which no oxygen was available. So oxygen is actually not going to play a factor into this first phase, our glycolysis, and not even needing or requiring a mitochondria. We are going to get a net gain of 2 ATP and additional energy molecules referred to as NADH. Additionally, what we're going to be getting out of some of this is going to be some water molecules. Again, this is going to be no oxygen required. So there are two stages that are going to be taking place in this glycolysis. Stage 1, energy investment. You have to spend money to make money, right? So we need to invest a little bit of our ATP to get some ATP out. So we have to invest some of this using our phosphorylate compounds. We are then going to do our phosphorylation, transferring that phosphate group. Stage 2, we are going to get the payoff. So once we spent our money, now we're going to make our money. Ultimately, we're going to net, again, not gross, but net our 2 ATP. Additionally, this is going to help us then reduce our NAD plus to make it an NADH, an actual energy molecule. Now, there are a lot of different steps that are going on in here. Please, 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 this is one of those things. Do not memorize this. If you're ever going to have any sorts of questions regarding this, you will be given these diagrams. But basically, there are these 10 phases that are going on in the process of glycolysis. Here's the top phase showing the energy investment. Notice how our ATP molecules, they are the zigzaggy, spiky bits. Again, that's how we know that they're energy are going into, and then ADP, the unenergized form, is coming out at various phases. Then, once we get to our phase number 2, energy payoff, now we start to get some of that energy back. So even though we had to give an initial investment of 2 ATP, we are going to get a gross of 4 ATP, but a net of 2. So make sure you know the difference between those two. Specifically in AP questions, they might say gross, which means just the total amount, and net. You have to take out any sort of investments or negatives to your total value. So be very careful about that. Now, in summary, here we're going to break it down, and this is mostly what we're going to focus on, is that we're starting with our glucose molecule, that's our sugar, and we're going to split it apart. We're going to lyse it into specifically these 2 pyruvic acid molecules, or pyruvates. Now, if you notice that these are 3 carbon-containing molecules. Now, our original glucose was 6 carbon-containing, so we are literally splitting it in half into 2 separate 3 carbon-containing molecules. And we're going to also get, or net here, some ATP and NADH. Additionally, we're going to have a little bit of our water being produced at this part, but not the bulk of our water. It's actually going to come from a different process. All right, so that's glycolysis again in a nutshell. Please do not feel as though you have to memorize all 10 of those phases. Be able to read and interpret the diagrams, that's important, but the splitting apart of our water molecules and netting those 2 energy, our ATP, as well as our NADH. Phase number 2, now you notice that we are actually going to move internally. So now we have to move across that membrane of the mitochondria to get inside so that we can continue on to our phase 2, the pyruvate oxidation, that's like 2.1, and then citric acid cycle 2.2, if you will, kind of splitting it into 2 different halves. So this inner space is going to be referred to as a matrix that's inside. This is kind of like an open space, jelly-like fluid substance that's going on inside of our mitochondria. And notice how the mitochondria has that double membrane layer. So it has the outer membrane as well as an inner membrane. That inner membrane, this is where our last and third phase is going to take place, the electron transport chain. That is where that's going to be taking place. Notice how additionally inside of this, folds on folds on folds, don't forget surface area is going to be increased with all of these folds going on, thus allowing us to then do our chemical reactions, or the ETC specifically, at a much more efficient rate. Just thought a little friendly reminder. So here's our phase number 2. Again, please do not memorize this diagram and be able to, regurgitate it kind of thing, but be able to read and interpret this sort of diagram. Follow the arrows very carefully. So if we see, here's our pyruvates, here's our cytosol on the outside, and then they need to move across the membrane. You may be thinking to yourself, wait a minute, we talked about things moving across membranes. I don't think that one can go across, just diffuse by itself, and you would be correct in that thinking. And so we do have to have a form of active transport is actually going to be required for pyruvate to then move across the membrane to get internally inside of the mitochondria. And that's something we'll come back to at the end when we play our numbers game and talk about how much energy in total are we getting. This is a little bit where energy numbers could then fluctuate. So we'll come back to that in a little bit later, but just keep that in mind that those pyruvates do have to move across that membrane, and it is going to require just a little bit more energy for that to take place. Now, additionally, once we get in here, here is our pyruvate oxidation, if you will. This is the 2.1, the first half of our second phase. So notice how we have our pyruvate is going to be converted into this thing called an acetyl-CoA. We're going to dump off one of our carbon dioxides in this portion of it right here, one of our products, if you will. Additionally, what we're going to do is we're going to produce another NADH molecule. This is another one of our energy carriers that we're going to save for the electron transport. Once this acetyl-CoA is then formed, it is then dumped into this citric acid cycle, this cyclic process right here, which the reason it's referred to as citric acid, you can imagine, is that we're going to begin with citric acid is going to be the starting. So at this part right here, we're going to start with a citric acid type molecule. It's going to go around once for each of our pyruvates. And this is part of where AP loves to play numbers games, so be very careful. If we have 2 molecules per 1 glucose here, how many times around would it go for the citric acid cycle? It goes around once per 1 pyruvate. So if there are 2 pyruvates, then it's going to go around twice for every 1 glucose. So be very careful. AP loves to play numbers games at this point. Additionally, when going around in this, we have a few different things that are going on. Again, follow the arrows very carefully. We have some carbon dioxide additionally is going to be produced, some 2 molecules of carbon dioxide in total, thus, again, going around twice. We have 3 NADH molecules, also including some additional releasing of our hydrogen molecules. We also have a little bit of ATP being produced and another one of our energy molecules. I know there's lots of them, FADH2. And we're going to stop there. That's the last type of energy molecule we'll talk about for a little while. So this, again, in a nutshell, is what's going on at our kind of overshell of our phase 2 here. So here's the first part. We'll break them down just kind of individually. So we have our pyruvate, again, is being converted into this acetyl-CoA. We're going to dump off one of our carbons. Specifically, it's going to be forming carbon dioxide. Additionally, we're going to get some NADH, one of our energy molecules. Notice how this NADH is produced inside of the mitochondria, not outside the mitochondria. So this one does not need to move across the membrane to be able to get to our electron transport at the very end. Here is our second of 2 phases of our number 2, which is going to be the citric acid, or sometimes referred to as the Krebs cycle. Now, what's going on with this is that our acetyl-CoA, once it's after gone through the pyruvate oxidation, then it's going to dump into the cycle, starting with our citrate, and additional carbon dioxide molecules are going to be released. So those carbons are slowly being peeled away, and then energy is being stripped of that glucose to pyruvate, to acetyl-CoA, to citrate, right? So this whole process that keeps going on here. Additionally, when it goes around, we're going to produce a net gain. So we're going to subtract out any sort of energy investments, if you will. We have a net gain of our 2 ATP, 6 NADH, and 2 FADH2s. Again, this is for 1 molecule of our glucose, because again, it goes around twice. So specifically at this location, this is what's referred to as the substrate level phosphorylation. This is not our chemiosmosis ATP production. So we want to differentiate between the 2 types of ATP production. This is substrate level, so substrate and enzyme, that sort of thing. Here are all the different phases. This is another one of those ones. Please, please, please do not think that you have to memorize this. So here is our acetyl-CoA being fed in, and then we're going through the 8 different phases, ultimately resulting in some of our energy molecules, 3 NADH per turn, and 1 FADH2 per turn, and 1 ATP per turn. But again, don't forget, ATP loves to play, or AP Bio loves to play numbers games. So this is just per 1 pyruvate. This is going to go around a second time because, again, in 1 glucose, there are 2 pyruvates that are then produced. I cannot stress that enough. So here it is, kind of a summarization, if you will. 2 pyruvates being fed, 2 acetyl-CoAs being produced and fed into the citric acid cycle, netting us. Here's all of our energy molecules, as well as carbon dioxide that is going to be produced. There's our 6 CO2, if you will. Last but not least, here's our third phase, the oxidative phosphorylation. We kind of talked about this in the last PowerPoint slide or last note section, so we'll kind of go over this a little bit more briefly. But as a reminder, we have the electron transport. This is where these these electrons are going to be dumped off. So now they are then being oxidized, if you will, because again, oxidation means loss of electrons. So they're going to release those electrons. Ultimately, those electrons are going to fall in energy, if you will, down to our final electron acceptor, which is going to be our oxygen or the O2 molecule. The big takeaway here is that when those NADH and FADH2 molecules are going to go through the system, that's the electron transport portion of this, that they are dumping off those hydrogens. So we're increasing the concentration of our H plus ion, aka reducing the pH. So pH is being driven lower and lower internally here. This is the inside of our mitochondria. The membrane versus the outside. This is the matrix over here. And then we are then going to use this high concentration of hydrogens to then file through our enzyme ATP synthase. This is going to mechanically drive or phosphorylate here our ADP to produce our ATP. This is known as our chemiosmosis, our second form of ATP production. And this right here is the moneymaker. This is where we get the bulk of our ATP energy. It's from our chemiosmosis, from all the NADH and FADH2 molecules being produced. So I know there was a lot going on here. You might need to, we'll go through this a couple times, might take you a moment or two to kind of go back and forth between this, but this does a pretty good job of kind of summarizing each one of our different processes, what's being produced, where are they being fed into. This does a good job kind of summarizing everything. Here's our 3 different phases. Phase 1 with glycolysis, producing our NADH as well as our ATP. Now, these NADH molecules, they do need to move across that membrane. Once again, just like these pyruvates, they do need to move across the membrane, which is going to be a form of active transport. So you are going to have to invest a little bit of money to be able to, or energy in this case, to move across that membrane. So you have to be very careful about that. And so that's why oftentimes when you estimate, well, how much energy or ATP in total are we making out of this whole process here? They usually give you a range. Anywhere from 30 to 32 at this end portion, 26 to 28 for this specifically. In part, what they're doing is that they're factoring in the active transport for these molecules, the 2 pyruvates as well as the 2 NADH molecules, to move across the membrane. So they're factoring in that we had to spend a little bit of energy just to move those things across the membrane. And that's why we see some range. So sometimes textbooks as well as scientists, they can't entirely agree. Like, what's the final answer? We give it a bit of a range because it depends on whether you're talking about things moving across the membrane or you're not. Okay, that's the whole process right there. The additional portion of this is going to be this fermentation. There are 2 different types of fermentation that can go on here. These are going to be without oxygen. So we, as oxygen-breathing organisms here, we need that oxygen so that we can do that end result, that electron transport moving into our chemiosmosis. We need oxygen to be able to do that, to make the bulk of our energy. If we did not, we would not be able to survive because we could not keep up on our body's energy needs. However, there are other organisms that can use different electron acceptors besides oxygen as their final electron acceptors. Or there are other things that can use sulfates, nitrates, or other sulfur-containing compounds. And a good example of this would be what's called an obligate anaerobe, which means it cannot survive in oxygen. In fact, if it were exposed to oxygen, it would die, which seems kind of counterintuitive if you think about it. I mean, from our perspective anyway, like, we need oxygen to live, but they would die in oxygen, right? So it's kind of a weird thing. The main mode of other organisms, though, would be what's called facultative anaerobes. So they have the ability, they can still live in oxygen, they just don't need it. They're not going to use it to process. So they're going to do these anaerobic processes, or without oxygen. So they can do this fermentation process. So they're not going to get to our final phase, the electron transport, because they don't have a final electron acceptor. Additionally, us as humans, we technically do this as well, this anaerobic process. This, if you think of when you are exercising and working out, when we are using our muscles and burning all that energy, when you feel the burning sensation, that is what's called lactic acid that's building up. You may have heard that, for you athletes out there, that our bodies, because we are so depleted of oxygen, because we're working out, we're breathing heavy, we're breathing faster, our heart's pumping right, our body's trying to get oxygen to every square inch of our body as much as humanly possible, because we're using all that oxygen to do cell respiration, to get the energy, because we're exercising, we're working out, right? And so as we begin to do that, why our body responds that way is that our oxygen levels are slowly being used up. Our reserves are being depleted. And at that point, then we begin to switch over from an aerobic process using oxygen to anaerobic process, which is going to be referred to fermentation in this case. Now, fermentation additionally can also be done by organisms like yeast, for instance. So it can produce ethanol, or the drinkable variety of alcohol. Please, bit of a side note here. Just because something says it's an alcohol does not mean it's a drinkable variety. Ethanol specifically is the drinkable variety that you would find in wine, beer, spirits, etc., right? But for instance, wood grain alcohol, yeah, it says it's alcohol. It is not drinkable. In fact, it will make you go blind. Is a prime example of that. Or rubbing alcohol, again, says it's alcohol, not drinkable. You will get violently ill if you try to drink rubbing alcohol. So please be wary about things that say alcohol. They are not all drinkable variety. Okay, here's our two processes again, kind of comparing between the two of them, with and without oxygen. So we have aerobic with oxygen, and anaerobic, which means without oxygen. We're doing the fermentation process. So we still have glycolysis going on in both of them, because again, notice how oxygen was not necessarily part of this. And again, those early organisms, they didn't have mitochondria. They only had that internal structure, that cytosol or cytoplasm here. And so they can only do that glycolysis, which is not very much energy yield. It's about 2, right? So it's not very much. It's not very good. Our yeast still do this today. We do it in our muscles, but our yeast is an organism that we then use to harvest, basically, to get the ethanol that they produce. Us as humans in our muscles, we do this fermentation process to produce lactate, which then becomes lactic acid, thus causing the quote unquote burning sensation in your muscles. Now, when oxygen is present, then we can do the full gauntlet. We can do all of those because we have that oxygen being the final electron acceptor. We can go on to do the electron transport, ultimately resulting in carbon dioxide, water, and our whopper of 32 ATP being produced. So oxygen, definitely to us, is very beneficial. Please keep breathing. And if you ever get super tired and winded and you do happen to have kind of those burning sensations, here's a bit of a side tip for you athletes out there. Take your hands, put them above your head, and try to take slow, deep breaths. So I get it, like if you're like on mile 2 or your 5K or something, you're probably like super winded and you're like, breathing slowly is probably the last thing you want to do. But honestly, try to take slow, deep breaths as much as possible, but definitely put your hands above your head. That opens up your airways just a little bit more so, and allows you to get a better breath. And so that's why we get that burning sensation. You kind of get the cramp. I usually get them on my side, is that your body's craving oxygen and you're not giving it enough. And so that's why we get that. As we mentioned, 2 types of fermentation: alcoholic fermentation done by bacteria, specifically yeast, to make wine, for instance, or even in baking, you can use baker's yeast to make bread and stuff. Additionally, here's what we do. We've been mentioning the lactic acid fermentation. We are only going to do the glycolysis portion, which is going to produce lactate ultimately because we don't have that oxygen being there. And then we build up the lactic acid in our muscles. It's also used to make other products, other types of dairy products like cheese, yogurt, acetone, which is nail polish remover, fun fact, and methanol, as I mentioned, that's wood grain alcohol. That is used to clean wood-based products. So we have additionally here, if you're wondering, so do we have to use glucose? Because what about like our fat? Technically, our fat is a storage of energy too, is it not? How can that feed into this process to help us make energy? So it doesn't just have to be glucose. Typically, when talking about cell respiration and the energy producer of our animal organisms, we typically start with sugars, aka glucose, for instance. That's the main supplier. However, we can't can use other sources, like, for instance, protein as well as fat. So if you don't happen to have those sugars and carbohydrates in your body will get it from other sources. So it will use the proteins that you are then ingesting and moving through your digestive tract, that's in your bloodstream and things. It will use that protein as well as fat reserves. So that fat can be broken down. It can go through some processes and enter into different phases. So again, follow the arrows very carefully, and you can kind of see where different sources, either proteins or fats, not just our glucose, our carbohydrates, can feed into this whole cell respiration process. Ultimately, they are going to go through basically the same phases, definitely going to end up at the citric acid cycle as well as the oxidative phosphorylation, are phases 2 and 3. So mostly they can enter in various phases at the beginning stages, not quite towards the end. There is one additional thing that is we want to talk about just a little bit, is going to be the inhibitors. So things that can then stop the process. So if you ever wondered, like, everything we've been talking about is how do we get stuff to go, 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 go? Well, what happens when you need to stop? Because we don't always want things to go. We don't always want things to just move forward. Sometimes we're like, okay, that's enough. We're okay. Our bodies need to then put a pause on some things. So we have these things that can do what's called inhibition, and then they can be stopped. So there are things that can push things to move forward or stimulate the chemical reactions, or things that can then inhibit and stop chemical reactions. In this case, there are products along the way that have the ability to feed back. This is called a feedback loop. This is something that we'll talk about in the coming weeks as we begin to dive into these processes a little bit more. Things that push things forward, stimulants, and things that are going to inhibit. So these feedback loops are either going to push the chemical reaction series to move more in a greater quantity, or to stop the process and slow it down. So just know that there are some control mechanisms if your body needs more, it can stimulate it and cause more, or if the body has enough of something, it can then have this feedback loop negatively here, inhibiting the process as well. So this is kind of, in a nutshell, all of the different processes going on. I'll pause here. Yeah, this is a pretty good job kind of summarizing this whole thing, beginning with our glucose. We're going to focus on feeding into our glycolysis and all the different parts that are needed. So hopefully this was helpful. Again, we are going to be working through this over the next week or so, really digging into this. So if you felt like a lot of this information was kind of a lot, you're not alone, and just know that we will be spending some time talking about it, discussing it, and going through the process to break it down and answer any and all questions you happen to have. Thank you so much for watching. Sorry this is a little bit longer of a recording. Please feel free to reach out to me or show up to class with questions. Thank you, everybody.